pluripotent cells and the differentiation process. So very, very quick introduction about me. I recently graduated from the U of A with a degree in biosci, and I joined the Wishart lab, working with the protein group into the CSIS lab. And lately, I've been working on the liver organ on chip project. So part of this liver organ on chip project is that we grow induced pluripotent stem cells, and we differentiate them into hepatocytes, which are then harvested and used in our models. So today for this presentation, I'm only focusing on the stem cells and how we differentiate them into hepatocytes, and won't really talk about much about the models downstream. So a bit about what I'm going to talk about, do a short introduction on what are induced pluripotent stem cells. I have a couple of videos I want to show. I was greatly inspired by Abby. So you can think of this presentation as like a part 2 to the cell culture tutorials. And then I'll also talk a bit about the differentiation process into hepatocytes, and then show one last video about how differentiation works. So first up, what are induced pluripotent stem cells, or as we like to abbreviate them as iPSCs? So first things first, to talk about iPSCs, you have to talk about stem cells. So stem cells are special cells, and they are unique from other cells in the sense that they have two very distinct abilities. First, the ability to self-renew, so that means they can replicate many times. They can replenish themselves. So in some organs, like in the intestine, the cells that line your intestine, or otherwise known as the epithelial cells, they're regenerated by a pool of stem cells. So when those epithelial cells die, new ones can be replenished. As well, stem cells have the ability to differentiate, so they can become more specialized cells with more specific characteristics and functions of the organ that they're becoming. And to explain this, they have different levels of potency. So potency is the capacity for a cell to differentiate into different cell types. So in this diagram, this is a very broad view diagram with different potency levels, but you start with a totipotency, which is when your cell is capable of becoming an entire organism by themselves. And what I mean by that is that they have the ability to become both embryonic tissue and also extra embryonic tissue like the placenta. If you go down a level or become slightly more differentiated, slightly more specialized, you reach the pluripotency level. I'll talk a bit more about that in the next slide. And if you go even further down level, you become multipotent. And these cells, the multipotent cells, are usually now more specialized into a specific organ or tissue type. And it can become multiple different types, cell types of that particular organ or tissue, but not necessarily become a cell type of a different organ or tissue. So in comparison, pluripotent stem cells are in that middle ground. They have the ability to become a variety of different cell types of different organs, but they can't become an entire organism by themselves. So they can't become the extra embryonic tissue like the placenta. Now, what we use is induced pluripotent stem cells. So they're not natural found pluripotent stem cells found in, say, a human. They're instead mature adult cells that have already been specialized into a specific organ or tissue type, and then they've been reprogrammed back to their pluripotent stage. And this is done by expressing certain transcription factors, which are specific to stem cells, including things like OCT3/4 or SOX2 and c-MYC and KIF4. Now, these cells have the ability to become any of the 3 germ layers, which are part of the embryonic tissue. So these include the endoderm, the ectoderm, and the mesoderm. We're most interested in the endoderm because that's where the hepatocytes come from. And you can see that in the cells circled in the pink circle. So what do the stem cells look like? Well, it's pretty cool. You'll see later in the video that these cells are different in the sense that we grow them as aggregates. A lot of times when you see pictures of cells, they're individual and by themselves. But here we grow them as clusters. And then when they attach to the plate that we coat with matrigel, which is a special extracellular matrix that supports the cell's growth, they expand and they become this kind of grainy looking colony. And then we can take these colonies, we can harvest them and do different downstream applications like differentiation. So next up is the video. I'm hoping this works. Can you hear it okay? Maybe not.
Cannot hear it properly. Probably you can try one more time.
Okay, sure. I mean, it's just music in the background, so it's not the end of the world.
Okay.
It will just be very awkward. I'm sorry about that. But yeah, I guess we can watch it in silence. So that's just a video showing what the different steps are of taking care of the stem cells. And now I'm going to talk a bit about the differentiation process. So like once you have the cells grown, how do you actually turn them into liver cells? So with the iPSCs, you have to differentiate them, which is a process to become a more specialized cell type, like discussed earlier. And this is usually done by changing how the gene expression and different signaling pathways are regulated. And essentially, there's a bunch of factors that are involved. However, from a more practical standpoint, it's just a matter of making sure you create the right cocktail of different growth factors, different, if there's cytokines or any proteins or hormones are added or supplemented to the media, and making sure that you make them at the right time and then change the media of the cells also at the right time, so that you're not pushing the cells too quick or making them go too slow down the differentiation pathway. So these are some pictures of what our stem cells look like when they're turning into hepatocytes. I've shown up here on the top figure a timeline of how many days it takes us to create these liver cells from the induced pluripotent stem cells and what each stage requires. So first, from day 0 to about a week, day 4 to 8, is when you grow your stem cells as aggregates, as shown in the first video. And then the later stages I'll show in the second video is where we'll passage them as single cells. So we're no longer growing them as the clusters that you saw earlier, but rather as individual. And then we change the plate that we grow the stem cells on. We change to a laminin-coated plate, and we add a Rho kinase inhibitor to the media to support the growth. And then we push the cells through their different stages until they eventually become liver cells. And you can see this reflected in the different morphologies in the figure below. And this is just a comparison of the results that we got to something from stem cell technologies who have developed the hepatocyte differentiation kit that we're using. And then now for the differentiation video. I'm just trying to get all of this video. And then that's basically the end of my presentation. I have a bunch of references here. The big bolded link at the top, it was like our holy grail for doing this project. It had a lot of resources and a lot of really clear protocols for when we were first trying to grow these stem cells. So if you're interested for more about how to grow stem cells, I highly recommend checking that manual out. And finally, a big thank you to both Dr. Wishart and Prashanthi for the opportunity to work on this project, and also to Upasana for showing me the ropes on how to grow cells, and also specifically stem cells as well. To Abby for giving all the tips and tricks for how to film a video, and also to all the other CSIS members and a bunch of other people as well. So thank you, and I'll also be happy to take some questions as well.